open modules. I never know where I should put it, but that seems to be the place that makes the most sense to me. Right? You have exam 1 review notes. So all of these chapter sheets for each of our chapters. There is a little video, a Pinocchio video of me drawing out the blood lineages on a whiteboard. So if you get confused about lymphoid and myeloid and who all the cells are and what their jobs, I will tell it to you again in very boring video format. And there are just some review questions that look an awful lot like the questions that are already on your study sheet, but maybe some other little additional things in here. So if you're going through this, you can use it like a checklist. This would be a really good thing for you to do this week. Use this like your checklist. Go through here. Be sure that none of these questions looks foreign to you. If there's some word, some term, something in here that you're like, what? Right? That, email me. Come here. Come to office hours. Come and see me to make sure that those terms make sense. I know that for a lot of us, taking tests in person, taking tests in a multiple format is kind of a new thing. We're getting back to doing it again, right? Because for a couple of years, it was like take home land, or everything was on Canvas, and right, it's just very different. I promise you, okay, that if you don't do as well on this exam that you would like, I will provide you opportunities to redeem the points that you would like to get. Right? All I really require to redeem the points that you would like to get is that you come see me and you talk about what you didn't do well on. Because my goal is for you to learn this stuff. Because somebody else is going to ask you other questions about these kinds of things. If you decide to go to graduate school, if you decide to become a PA, an MD, a veterinarian, someone else is going to ask you these things. I want to be sure you come out of here feeling confident that you at least know the basics of immunology. Me giving you an F didn't teach you anything. I want to teach you stuff. I want you to learn it and know it. That's my goal. So if this goes horribly wrong, you're nervous, you're right, something wasn't right, don't worry about it. The best thing you can do before a test, especially if you've studied and you know you're prepared, is don't worry about it. Right? Because anxiety is a person who has suffered from anxiety their whole lives. I get it, okay? Don't psych yourself out. You know what you need to know. But this is your checklist. This is how you know whether or not you know what you need to know, right? Use these as your checklist. Again, please, if anything, from any of this stuff, you're like, can we rewind and talk about chapter 1 things? Yes, we can. That's why we're here, so that we can review so that you know it. So that's it. Is there anything we would like to review? Right? Maybe you're beginning your review this weekend, you're going back, you're asking yourself, do I know what that term clonal selection means? Do I remember the difference between innate and adaptive immune cells, right? Some of the characteristics that differentiate innate immunity from adaptive immunity, right? I guarantee you there's going to be an exam question where I ask you to kind of compare and contrast the difference between how adaptive immune cells work or how innate immune cells respond to things. I know you haven't learned a whole heck of a lot about the adaptive immune response yet. I'm not looking for details, just generalizations. Okay, no questions. Are we sure? There's purple pieces of paper this week. If you have questions and you just want to write down, and I will answer them for you. There's some on that table, and there's some as you come in the door. All right, chapter 3 is all about signal transduction. Okay? It's important for us to remember that nothing happens on this planet without signal transduction. Hormones binding receptors, phagocytic cells crawling around in the world, how do they know what they're touching? How do they know to respond to what they're touching to? They use receptors on their surfaces. They contact ligands. That tells them something. That helps them change their behavior. That tells them do phagocytosis. That tells them keep on crawling. That's nothing. Okay? So that's the crux of this chapter. It's also, hint, sort of the crux of the next chapter as well. When we're talking about cytokines and chemokines, we're talking about the hormones and receptors that get immune cells and our tissues to do what they need to do to protect themselves from invaders or toxins. So, as long as we get the gist of this, you're good. There are some simple basic concepts for us to think about in signal transduction that are always true, right? A signal is an event that instructs a cell to change its behavior in some way. That could be a chemical trail that tells a monocyte it's time to leave circulation to crawl into a tissue to follow that chemical trail to go to the site of infection or trauma. Right? It could be a hormone. It could be a lot of different things. But whenever these chemicals bind onto, whenever the ligand binds onto its receptor, a series of events, a chain of events are going to happen inside the cell that get it to do something in response to that trigger. Ligands can be soluble molecules. They could also be another receptor, a membrane-bound molecule. It could be a peptide. It could be a carbohydrate. It could be a lipid. Right? There is no limit to what our cell surface receptors can sense. Ligands can travel long distances. Sometimes they only work locally in a small area. Depends on what's happening, depends on the context of the trigger. Right? We have some great examples of early on in inflammation. You get a cut, right? Scrape, a wound. This is a localized site of inflammation where things are only happening on a small scale in this little area of the tissue. But what if this is a great big cut? I'm bleeding a lot. This could send a systemic signal through my body, right? That eventually turns on the acute phase response for my liver. Send more complement. What if it's even bigger than that? What if lots of microbes get in here and they're causing an infection? That signal gets all the way through my body, flows all the way up to my pituitary gland and my hypothalamus and tells me to have a fever. Right? So things can be localized, they can be systemic. Generally, receptor ligand binding is non-covalent, so this is not a permanent interaction, but it can happen with very high affinity. I like to think about lock and key. Right? The shape of the receptor dictates what ligand will be capable of finding in there. And think about, right, all of the different chemical structures that exist on our planet, different hormones, right, the 4 major macromolecules. You have sets of receptors whose job it is to bind those specific things to trigger reactions to happen inside the cell. Often, ligand binding a receptor changes the receptor itself. Something happens to the receptor when the ligand binds. Sometimes that means the receptor autophosphorylates itself, right? That phosphate acts as a trigger, as a dock to recruit more activating proteins. Sometimes receptors dimerize. So when one binds the ligand, two come together. That causes signal transduction to happen. Sometimes binding the ligand causes something called lipid rafting. A whole bunch of things that are like lipids come and bind together and make this big giant thing on the surface of the cell that actually penetrates down through the plasma membrane into the cytoplasm of the cell. Can change the localization of a protein, sometimes being getting different types of covalent modifications. I put special types of fat molecules like myristoyl groups or palmitate groups. If a fat molecule gets added onto a soluble protein, it's no longer soluble anymore. It gets recruited to a membrane, so that brings it to some new location. All of these different modifications change where the proteins are, change their behavior, turns on enzymes. Often, the whole point of inducing signal transduction is to change the behavior of the cell. So the end result, how am I going to change the way a cell behaves? I have to turn on and activate genes. So turning on genes through transcription to get a new gene product made, right? Eventually, I'm going to take that RNA product from transcription, I'm going to translate it into a new protein, a hormone, a chemical signal that the cell will then use to do something, to protect itself, to increase inflammation in the environment. All sorts of different outcomes can happen. Right, here's our lock and key principle. Specific ligands bind specific receptors, always. Right? There's not just this universal, like, 4 receptors, and that's it. There's millions of receptors. When a receptor and a ligand bind, we're talking about, right, puzzle pieces, lock and a key. This means there's points of contact between the 2 objects, between the ligand and the receptor. And those points of contact, what kind of bonding do they involve? How many points of contact? These determine 2 terms that are pretty important. Affinity, right? Affinity is the tightness of the interaction. Avidity, A-V-I-D-I-T-Y, is number of points of contact. So you don't necessarily have to have super strong types of bonding happening. You don't have to have ionic or covalent bonding happening for a lot of mass action holding a molecule together. Great example, right? Hydrogen bonding holds the rungs of the DNA ladder together, of our DNA. Hydrogen bonding is a weak bonding force at best. But all the mass action, all of those hydrogen bonds, right, all along the rungs of the ladder, hold that molecule together. We're actually very fortunate that it's not ionic or not covalent bonding, because every time we'd have to open that ladder up, we would expend a lot of energy to do transcription or to do replication. Often, when a ligand binds a receptor, other receptor molecules get involved too. So co-receptors can help out. They can be partners in the reaction, and we especially see this in immunology. Groups of toll-like receptors work together to help increase signal transduction inside the cell. The B cell receptor, along with other co-receptors like B19 and a whole bunch of other accessory co-receptors help out. T cell receptor does this too. You almost always have partners who help you in signal transduction to help increase the strength of the message entering the cell, to help the cell know what to do. So here's a nice example here where we remind ourselves about affinity and avidity. So multiple interactions is avidity. Right? The more points of contact I have, the stronger that interaction is. Doesn't necessarily have to be strong bonding forces, could be the mass action of all of those bonding forces working together helps. You could have a high affinity interaction, right? The strength, right? Just being able to let them stick or pull them apart like magnets, or how many points of contact. Both things help. Ligand and receptor expression can change over time. And this makes sense, right? The amount of hormones our body is secreting at specific times change, depending upon the stressors. But this also happens to receptors too. So this picture right here shows us white blood cells. These are going to be lymphocytes, not leukocytes. The reason why I know that is because they are responding to a particular cytokine called IL-2. Interleukin 2 is a member of the hematopoietin cytokine family that we're going to talk about in a minute. But this is a growth hormone that pumps up B cells and T cells. They love it. It's their favorite growth hormone. So we have some T cells hanging out in the environment. You can see another characteristic thing about them. They have gigantor nucleus, but relatively small little sliver of cytoplasm around them. That's very stereotypical lymphocyte. Giant nucleus, small cell altogether. If I feed these cells interleukin 2, they have some interleukin 2 receptors on their surface already. They're waiting for that signal. But if I feed them interleukin 2 and I stain them for the amount of interleukin 2 they're making, see they all start glowing bright yellow everywhere. It's because you feed them a little bit of it and they get super jazzed up and they send all of their internal receptors to the cell surface so that they can bind as much interleukin 2 as they can. They're very jazzed up about that hormone. They want all of it. Makes them very excited, gets them activated to grow and proliferate, to grow and divide. This is what gets clonal selection and expansion going. So in responses to particular triggers, our bodies become more receptive to those signals. This happens with immune cells, it happens with our other tissues as well. The more exercise you do, the more glucose transporters get transported to the surface of your cells to help you bring more fuel in to keep your muscles working, right? So you get these sometimes positive feedback loops. The opposite can happen as well. Sometimes a ligand binds a receptor, and that makes our cells downregulate that receptor, do the opposite response. So we can have positive and negative feedback loops depending upon what ligand and what receptor are involved. Local concentrations of cytokines at the site of trauma, at the site of infection, might be high. So what happens is the cell can actually direct its attack. It's going to sense chemical gradients using its receptors, and it can make sure that it directs its attack in a pointed way. There's an example right here that we have blue dendritic cells that are doing antigen presentation and trying to activate a T cell partner. So these are the dendritic cells, these guys. Here's my T cell partner. My dendritic cell is using cell surface receptor interaction to interface with this T cell. So you can think about it like they're holding hands. They're in very close contact here, so dendritic cell knows, oh, my T cell buddy is right here, and I need to provide my T cell partner information. So what's happening right here is this dendritic cell is going to take this packet of cytokine, of IL-12, and deliver it directly on to the T cell. So he's not just going like, oh, I'm just going to send IL-12 out anywhere. I'm going to deliver it directly to my partner. That's what these points of contact tell the two cells. We're holding hands right here. I'm going to deliver the package to you directly to get the outcome that I wanted. This is up close, super duper close, so that you can see the space in between the two cells, and you can see how this packet of information is being delivered directly to the cell. Instead of just, no, I'm just going to send that out, and you can pick it up, I'm giving it to you on purpose. What's going to happen when antigens bind to receptors, like toll-like receptors, or selected type receptors, or a T cell receptor, or a B cell receptor. What happens? We turn on, right, an internal signaling cascade. And this is another key point to remember. Signal transduction happens inside cells. There are hormone cascades and proteins that get activated externally to our cells, things like complement, but they're technically not doing the same thing as what we're doing here. When this happens, what are some of the changes that can occur? Motility, changes in motility in cells. I'm going to sense a chemoattractant, and I'm going to keep crawling towards it. So I'm going to change my motility. That's what chemokines are all about. There is one whole family of cytokines whose job it is to make cells move. Change their adhesive properties, get cells to hold on to each other, stick tightly, interact, or make them let go. Turn on changes in transcription. Get me to make new gene products. Hormones so that I can protect myself. Hormones that I can send out into the environment to warn others or to protect others. Here's just a generalized example of signal transduction. I have my main receptor right here. My ligand is binding into some kind of receptor binding pocket. We have both affinity and avidity interactions to think about here. There is a co-receptor involved. So perhaps this could be like a complement receptor. This ligand is a bacterial carbohydrate, and a complement protein has bound onto that too, and now this co-receptor is binding onto that bound complement protein. All of these receptors involved, right, act as docks. They're going to form lipid rafts. They're going to autophosphorylate. They're going to dimerize, which makes all these cool little phosphorylation events happen on the receptors themselves. Those phosphorylation events, right, recruit new proteins, adapters and dock proteins. Usually what happens with adapters is they plug on and they act as a docking station to recruit other proteins. So the adapter gets bound, it sees those phosphates, it sticks onto them, likes that. Ooh, good charge. Once that happens, the adapter itself gets phosphorylated. And it's going to, in turn, recruit new partners. Right? So delivering all these signaling molecules to where they need to be to become activated. Some things are going to physically change their location. We're going to break apart different phospholipids and change them into different secondary signaling products. We are going to activate GDP, GTP exchange factors. Those are often very important in chemotaxis, in movement, getting polymerization of actin so that cells can crawl and move. We are going to turn on all sorts of secondary signaling messengers. We might do something like release intracellular calcium from our endoplasmic reticulum out into the cytoplasm. Calcium's going to bind onto calmodulin, get it activated. Now these little pieces are going to stick together and they become a transcription factor that is active and can enter the nucleus and activate particular genes. So sometimes the goal is movement, sometimes the goal is actual transcription, getting you to make a new gene product to get you to do all sorts of different things. These are just some examples of very common signal transduction pathways that are involved in lots of different processes. The PKC pathway activates NF-kappa B. The RAS MAP kinase pathway is often very important in growth and proliferation. The PI3 kinase and phosphoinositide pathways are also very important in regulation of metabolism, growth, and proliferation. These all have lots of diverse, different functions, different genes that we turn on that get cells to do something in response to a trigger. Here's an example where we're looking at a growth factor binding onto a receptor, causing its dimerization, causing autophosphorylation to happen to that receptor, allowing these, right, phosphates, their negative charge, to act as recruitment molecules to bring signal transduction machinery to the receptor. Yes?
I see RAS and MAP proteins and cascades everywhere. Are they always the same cascade,
Pretty much.
activated by different things?
Pretty much. There is, to make life even more difficult, there are probably about 20 different signaling proteins that slightly change whether the RAS pathway activates movement or whether the RAS pathway activates MAP kinases for growth and proliferation. So there are some little early on events that help us decide, do we move or do we activate transcription? But there's also crosstalk between these two pathways. So some of the MAP kinase enzymes can work with and phosphorylate members of the PI3 kinase family, and also activate and phosphorylate some of the members of the NF-kappa B pathway. So these can be ways where individual signals come through and tell us all to do one thing, but the signals can all meet at particular traffic points, and when they do, that turns on different responses. How many pathways get turned on at the same time? That also tells the cell different cues about what to do. So this is like, it's a mass action thing. How much stuff gets turned on? Who gets turned on? Helps the cell decide, helps it know. Right, so all of this, these early events, right? In this case, dimerization doesn't have to be, but this is an important one that can happen. Here's a great example. Here's my good old B cell receptor, BCR, right? A B cell receptor is an antibody that is anchored into a membrane. B cells can choose whether to anchor their BCRs into the membrane or whether to allow them to be soluble and excreted. But in this case, when a B cell is first born, all of its BCRs are membrane anchored. When it triggers an antigen, this can cause recruitment of lots of different B cell receptors to the same antigen. They can all be holding it at the same time. Co-receptors can also be binding here too. But you can see we get phosphorylation of the intramembrane receptor portions of the molecule. Phosphorylation recruits an adaptor protein, which is frequently BLNK or blink. And that guy acts as another recruiter to bring things like SYK or RAS or PI3 kinase or a whole bunch of other different things from happening. So it doesn't really matter, are you a cytokine receptor, a more generalized hormone receptor, are you an antigen binding receptor, a toll-like receptor, signal transduction is the outcome that's going to happen. So in lipid rafting, usually what happens is a whole bunch of types of receptors bind together into a giant conglomerate, all holding on to generally something that would be a larger antigen. Here's another example of a co-receptor getting involved. In this case, it's CD21, which recognizes a particular complement molecule. So I've got antigen, it's being bound by the B cell receptor. It's also been opsonized with complement, so complement receptors help out too. This is a T cell receptor. T cell receptor complex is a little more complicated than the B cell receptor complex. You've got the binding portion of the molecule itself, but then you have all these little accessory co-anchors that help hold the TCR into the membrane. TCRs always interact with another receptor. They can't bind naked antigen on their own. They always have antigen presented to them by some type of MHC molecule from an antigen-presenting cell. So this is a dendritic cell, or a macrophage, or a monocytes, delivering this cargo, showing it to the T cell, T cell, and this is what's really cool, a T cell recognizes both the receptor, like the hand that is delivering the molecule, as well as what is the molecule inside the hand. So T cells pick up important information from handshakes. They look at the hand. They also look at what is inside the hand while they're doing the handshake. There's an accessory co-receptor that is critically important in activating T cells. This thing is called CD28. One handshake is not enough. We need multiple handshakes to know what's going on, to convey information. Because we don't want T cells to just get excited and run around killing stuff. That would not be good, right? We don't want them to just get excited and secrete inflammatory hormones and create inflammation where we don't want it. We want multiple points of contact, multiple receptor interactions, helping guide the T cell to know what the appropriate response is. Tyrosine phosphorylation, serine phosphorylation, threonine phosphorylations, right, are critical events in triggering signal transduction cascades. So you've got all of these R side chains on amino acids, which have OH groups. Right, those O's allow for bonding. Right, often those events are going to be phosphorylations, but we can phosphorylate tyrosine, serine, threonine. Those are very common events that trigger signal transduction. Yes?
On that past slide, what was the top cell?
So, so this is our T cell blue membrane, right, TCR. This top piece, yellow membrane, most likely this is a dendritic cell, a monocyte, or a macrophage. And the way that I know that is that they're using this molecule, right, it's labeled class 2 MHC. Only a handful of special cells in our body make class 2 MHC. They are professional antigen presentation cells. So monocytes, macrophages, dendritic cells are professional antigen presentation cells. So that's how I know this is some kind of professional APC, as opposed to some other cell that could accidentally or, by the way, be doing antigen presentation. It would do it using a different receptor, not this one. We're going to get there. You'll learn all about class 1, class 2, how to tell the difference between what you're looking at in these receptors, but that's after we take these tests. Okay, so phosphorylation events, right, are going to be often the trigger that turns on signal transduction. In this case, they use a SARC family of tyrosine kinases. The SARC tyrosine kinases are members of the MAP kinase family. This points out again, right, what residues often get phosphorylated, serine, threonine, tyrosine, right, because they have hydroxyl R groups. Pull off that H, now you got an O. O is all about covalent bonds, right, sharing 2 electrons to satisfy that outer electron shell. Phosphate is all about it too. So we get phosphorylation events happening here. This can turn on an enzyme. It can also turn off an enzyme. We usually think about it as a stimulatory event, but sometimes it turns things off. Can induce the interaction of 2 proteins together, making changes in conformation or appropriate charges, change the location of a molecule, making it move to a more hydrophobic or more hydrophilic region of the cell or to a membrane or not, help protect a protein from destruction, or trigger the protein for destruction. So phosphorylation can activate or it can turn off, but it can also preserve a thing or mark a thing for destruction too. All depends on the protein that we're talking about in the signal transduction pathway. Here's some other examples of adapter features that are going to recruit proteins to a site where a thing is. So SARC homology 2 domains that recruit members of the SARC homology family are always phosphotyrosines. SH3 domains are another domain that we see, but these are proline-rich sequences. So these prolines, right? Prolines have a weird sort of turn, and then they change the folding. They change the secondary and tertiary structure of a protein. There are proteins that correspondingly have phosphotyrosine binding domains. And whenever something gets phosphorylated, tyrosine gets phosphorylated, there are proteins that are attracted to that. All sorts of different types of motifs here. Here's an example of one that changes the location of where a protein would be. So we got a little protein floating around in the cytoplasm. It's mainly soluble, right? Cytoplasm is mostly liquid. I get myristylated. I get 1433 marker put on me. Myristylated, right? Myristyl group is a 14 carbon long chain, right? Hydrocarbon. Long chain hydrocarbon does not want to be hanging out in an aqueous environment floating around in the cytoplasm. So now this protein is going to be delivered over to a membrane, and that myristyl group is going to insert itself into the plasma membrane because it hates being soluble. So now we're changing the location of that molecule because of the modification that happens to it. There are a lot more different types of domains that exist than what are listed here, but these are some of the most common ones that trigger signal transduction. There are often adapter proteins that are going to help in these processes. So phosphorylation of lipid membranes, for instance, is going to recruit a new domain-containing molecule to that membrane, which in turn will activate new chemistry, new signal transduction. So a great example here is shown when we look at, this is phosphoinositide phosphate 2, PIP2, hangs out in the membrane. And when the signal transduction pathways get turned on, often an enzyme called PI3 kinase, this thing, phosphatidylinositol 3 kinase, gets activated, and it comes and it phosphorylates this PIP2, turning it into PIP3, which will now recruit other plextran homology containing proteins or enzymes to this residue stuck in the membrane. Okay, just some of the things that happen that trigger signal transduction. Here's some more close chemistry, looking at an additional chemical reaction that happens when PI3 kinases are activated. So we're generating PIP2 and PIP3 in the membrane. This recruits an enzyme called phospholipase C. Phospholipase C comes along and cleaves PIP2, releasing these inositides, which act as secondary messengers in signal transduction. These guys, this molecule right here, binds onto the endoplasmic reticulum of cells, and when it does, it opens store-operated calcium channels. So this releases calcium into the cytoplasm. That calcium is going to bind onto calcium-responsive proteins or enzymes in the environment and get them activated to do a job. Right? So like it's a domino effect all the way down the line for signal transduction. Right, so here's our good old calcium calmodulin. There's a closer look at PLC and PKC. So activation of phospholipase C and generation of phosphoinositides further activates this thing called protein kinase C. Protein kinase C does its work to help release a transcription factor that's going to activate genes inside the cell. Here's an example of RAS kinases. In this case, we're looking at RAS as it works as a G protein. So RAS itself, right, is a guanine nucleotide exchange factor. It works with GDP to become active, to cleave, change something, right? It then exchanges, it hands GDP away and becomes inactive. So it's going to use an energy-bearing molecule to do a job. So RAS, right, binds GTP. It stimulates its breakdown to GDP, but while it's active, it is going to do something to some target molecule. While GTP is bound to trigger signal transduction. One of the common things that can be activated is a further signal transduction pathway that activates the transcription factor called AP1. But another thing that can also happen is movement of the cell, and I think I have a picture of that in the next one. But here's my RAS being activated. It recruits this adapter protein called SOS, SOS, SOS7-less. Once that guy is activated, it activates RAF. Once RAF is activated, it activates MEK. Once MEK is activated, it activates ERK. And once ERK is activated, ERK becomes a transcription factor, so it can now leave once it gets phosphorylated. If it's not phosphorylated, it's trapped in the cytoplasm. Once it gets phosphorylated, now it can cross. It can bind into the nuclear pore, it can cross into the nucleus and act as a transcription factor. You'll notice here that right here it says this is a MAP kinase. This is a MAP kinase. This is a MAP kinase. Right? Again, another place where I'm a kinase that phosphorylates a kinase that phosphorylates another kinase, so we're on the MAP kinase. Dumb. Really, very dumb. Not good naming, but you get the point. Signal transduction moves through the cytoplasm to activate a transcription factor, which turns on genes. Gets the cell to change its behavior. Right? So here's phospho-ERK. Phospho-ERK then, right, can leave the cytoplasm, enter the nucleus, activate ELK1. ELK1 goes and it turns on genes. So this is my really awful, this made me laugh really, really hard, because when I was a graduate student and I entered school, I studied RAS-MAP kinase pathways. My first publication was actually about MEK and ERK, and my whole life I've dealt with MAP kinases, and this made me laugh so hard because I'm the kinase that phosphorylates, right? Your kinase and your other kinase. I phosphorylate, your phosphorylation. It's pretty dumb, but if you're me, it's hilarious because you spent your whole life studying this. You get up at 2 in the morning to go lyse cells to extract cytoplasm and figure out if MEK or ERK gets phosphorylated at a particular time. Another very cool pathway that is ubiquitously activated in cell regulation is the NF-kappa B pathway. This stands for nuclear factor kappa beta. They are transcription factors that are trapped in the cytoplasm unless the tethering molecules that keep them in the cytoplasm get phosphorylated and destroyed. So this is an example where phosphorylating one of these products actually allows it to be destroyed, which then releases I-kappa beta, which then allows it to become an active transcription factor.
So is that like negative feedback?
It kind of is. I don't know if I'd truly call it a negative feedback because in this case it's just the phosphorylation of this thing marks it for destruction through NEMO and ubiquitination. It's not truly like, it's not turning off the pathway, it's just removing a regulatory protein. Yeah?
You said something about labeling pictures or whatever earlier.
No, oh no way. I would never ask anyone to label a signal transduction.
Well, I mean, I had to do it in.
I
biochem,
bet.
so-
Yeah, I bet some like some
Cell biology.
sadist, right? As a person who worked on this for like 8 years of my life, there's no way I would force any other human to label a signal transduction diagram. What might I ask you to label? I hint, I'm leaning towards giving you a diagram of hematopoiesis. I'm going to give you a word bank so that you know what kind of terms I'm looking for you to label. But I'm looking for generalized stuff. Do you know what the difference between the agranulocytes and the granulocytes are? Can you categorize the cells into the right group? Are they innate or are they adaptive? Do you know where they come from in the lineage? That might be a thing I might ask you to label. I might, if I gave you something like this, a signal transduction pathway to label, I might say, I'd give you something very generic, right? And I'd say, okay, a ligand has bound a receptor. Tell me what's going to happen to the receptor, right? And I give you a little picture like a phosphate, right? You draw a little P on there, and then who does that recruit, or whatever. It would be very generic. Be like, tell me which one is a receptor. Tell me which one is a ligand. Tell me who's an adapter protein. Tell me what are these special molecules that bind on DNA and activate genes. What are those called? Something like that. Basic. Simpler. That's just mean. Straight up mean. Here's NF-kappa again, right? So these guys, one guy gets phosphorylated, then it gets chewed up. This guy gets phosphorylated, it gets chewed up, and NF-kappa gets released to go do its transcription factor stuff. Just examples of signal transduction, right? Hopefully, are we beating the horse enough? It's dead. It's totally dead, right? You've seen the ligand's going to bind a receptor. It's going to cause something fun to happen to the receptor. The receptor is going to autophosphorylate or dimerize. That's going to recruit an adaptive protein. That's going to trigger signals to move along the pathway to release a transcription factor to activate genes. That's it. If you know that that's what signal transduction does, that's really all you need to know about signal transduction, because that's the point. It's the point of activating any signal transduction pathway. Why are there so many different signal transduction pathways? Do you have an idea why there are so many? Yes, redundancy. Redundancy. I have to have a way to be able to get information outside the cell, inside the cell. Also, right, there's potentially lots of different ligands, lots of different signals coming in. So I need to be able to tell the difference between food, foe, hormones, right? So some of that is going to be pieced out in which signal transduction pathway or which group of signal transduction pathways are activated with a given signal. But really, right, this is how cells know how to respond. So I was talking to someone the other day, and they were asking me about, like, really, so if a macrophage is crawling around, it's not just the surface, right, the plasma membrane of the cell that's touching another cell that lets the macrophage know what it's doing. Correct, it's receptors. Every surface of our cells are coated with these little hands sticking out of them. Do the hands hold on to each other? Does something happen, or do the hands just slide on by each other? Is the way that this is happening. I'm going to show you a picture. I'm choose wrong first. It's chapter, yeah. This is my favorite picture to illustrate this. Complement, right, is opsonizing a pathogen. All 3 complement pathways have been activated to opsonize this pathogen. How does opsonization get my phagocyte to be excited to do phagocytosis? You can see, right, my phagocyte has all these special little cell surface receptors, and they're different. This one binds onto this thing, this one binds onto a little purple dot, this one binds onto antibody, this one binds onto this weird, whatever that is, right? Cells are covered, decorated in these special sensory molecules for knowing what to grab onto, what to eat, where to go. All of that, right, is triggered by signal transduction. Every stupid thing on the planet is triggered by signal transduction. Okay, so this last bit in the chapter is giving you a look at the T cell receptor and the B cell receptor and understanding how those receptors help B cells and T cells specifically to see unique antigens. Right? We know how complement receptors work to see things decorated with complement. We know how toll-like receptors, right, respond to some things extracellularly or intracellularly. C-like leptin receptors, right, RIG receptors. There's all these families, right, that bind to molecular patterns. The PCR and the TCR don't bind to patterns. They bind to unique features on antigens that maybe pattern recognition receptors wouldn't see. Right? This is where their secret power lies. They have the ability to recognize things that aren't patterns. Because pathogens want to disguise themselves. They don't want to be seen. They may hide or protect their patterns so that they can evade detection by those cells. Here is the family of receptors that belong to what are called the Ig family. Ig stands for immunoglobulin. An antibody is an immunoglobulin. So you're often going to see in scientific reading in texts, we abbreviate antibody, but you're also going to see Ig. That means immunoglobulin. It's the same thing. Immunoglobulin, antibody, same thing. So this is the immunoglobulin family of receptors. Here's the antibody. Looks just like the letter Y. 2 little feet plant the antibody into the membrane sometimes. You got 2 little arms coming off the hands. Right? Bind antigens. So antigens bind right up here in these little pockets. Those are the antigen binding pockets. Here's a T cell receptor. It looks kind of the same. Its arms, right, aren't as far apart, aren't as spaced apart. It's almost like a T cell receptor. It's just this little end bit. It's just a hand plugged into the membrane. But this is where antigens bind in the T cell receptor. These are MHC receptors. These are the molecules that hold antigens and plug in to the T cell receptor. Notice they all have these kind of similar shapes. There's 2 stalks, and then stuff gets bound in between the 2 stalks. There's a little binding pocket. These guys also belong to the immunoglobulin superfamily. The reason why they do is they have essentially, if I cut a TCR in half or I cut a BCR in half, you'd have one of the chains that looks like this. So these are the ICAMs, intracellular adhesion molecules. These are the molecules that plug together in our tissues to make organized muscle fibers, organized liver, organized whatever organ, right? It's all plugged together in fancy, nice layers, organized neatly because of adhesion molecules, right? So there are VCAMs and ICAMs with a whole deal is plugging cells together in layers so that they